biology students. Today was the first day that we did our microscope labs in class, and if you're remote learning, you used a virtual microscope. I hope that went well. You still do need to answer the worksheet questions, so I thought I would show you some things that I had on the board today with our on-campus students. So the first thing here is I want you to see how you find the magnification of the microscope. Now I know everything is backwards, and I'm sorry about that. But what you do is you multiply the eyepiece lens, which is always 10. That's the part you look through, by the total by the objective lens. I'm sorry. So the objective lens, like low power, is 4. Medium power is 10, and high power, the long lens, is 40. So you multiply each objective lens by the eyepiece lens. So for example, low power, as you can see here, is 4 times 10 to get a total magnification of 40. Medium power is 10 on the objective lens times 10 on the eyepiece you look through to get 100 times magnified. And then high power is 40 on the objective lens times the 10 on the eyepiece lens to get you a total magnification of 400. So when you're drawing your sketches, you need to write below them the total magnification. So please use 40 for low. 100 you don't have to draw, but just so you know, that would be for medium, and then 400 times for high power. Okay, so one other thing on your drawings. So on your low power for your cheek cells, I know you're doing virtual in a lab, but your cheek cells should be only 40 times bigger than what you would see with your eye. So you just need to draw them in the circle on page 2 after you do your pre-lab questions. And then on your high power cheek cell, you need to draw at least one big cell. It looks kind of like a fried egg. You need to label 3 parts, and I want you to use different colors so I know you know what you're identifying. So again, I know it's backwards, but like the nucleus would be in the center. It's like the yolk of a fried egg. The outline of the egg is the cell membrane. And then all the gel that's inside that you may not really be able to see, but we dyed it blue in our lab on campus, that's the cytoplasm. So I hope that helps a little bit for the animal cell, that's your human cheek cell. And then for the onion, some classes didn't finish this one today, but if you're still working on it for day 2, you will need to draw your onion at low power. It looks kind of like a brick wall, but really when we saw it through the microscope, it looked more like snake skin or mesh or a net. So at 40 times in low power, you need to draw something like that. And then on high power, 400 times bigger, you have one extra part to label for the plant cell. So you have your exterior, which is the cell wall. Now the animal cells don't have that. The cell membrane is inside the cell wall. So I use different colors. Cytoplasm is the green color here. It's that jelly that surrounds all the organelles. And the only organelle that you have to list is the nucleus. Okay? So if you have questions, let me know. And I'll see you on Thursday for any other questions. Tomorrow we're going to wrap this up in class. If students need to, you do have a quiz over macromolecules and enzymes, so please look over your papers. And what else? Keep working on your lab, try to get it done, and if you didn't get everything done in your 45 minutes, I'm sorry, you might just have to do a little extra. So I'd be sending homework home for my in-person students. So do the best you can, ask for help if you need it. Let me know how it's going on Thursday or before. Bye everybody.